Welcome students. Now we will study in detail about mRNA. In the previous class we have said that RNA is of three types. RNA is of three types, that is mRNA, tRNA and rRNA. mRNA, it is also called as messenger RNA. It is also called as messenger RNA because it brings the code from DNA, which helps in the synthesis of protein. And then we have transfer RNA, that is it brings the amino acids from the cytoplasm to where the proteins will be synthesized. And the third is the ribosomal RNA, the place in the ribosome where the protein will be synthesized, that is ribosomal RNA where proteins will be synthesized. Now how that process will take place? How the process will take place? How the information from DNA is given to mRNA? How the mRNA translate the information and how tRNA transfer the information, and finally how proteins will be synthesized. All this we will be seeing in detail, because I already said you proteins are the basis which helps, proteins are the ones which helps in carrying out the various functions of our body. Without protein we cannot carry out any single function. So proteins are the basis on, that is by which the whole process will takes place, that is respiration, excretion, all these activities requires protein to, that is proteins for the cells to perform. The cells requires proteins. How proteins will be formed? It is because of RNA. RNA helps in the synthesis of proteins. So we have seen the type, that is what is DNA, how DNA is made up of, with what DNA is made up of. We have seen what is RNA made up of, that is both are nucleic acid made up of sugar, phosphate, and that is both are made up of pentose sugar, phosphate, that is phosphoric acid group, and nitrogenous base. Then we have already seen the three types of RNA. Now we will see in detail about how these three types of RNA helps in the synthesis of proteins. So we'll start up with the first type, that is the mRNA. mRNA is also called as the messenger RNA. Till now we have already seen it. Now let us see how this will be formed, that is how the code from DNA will be helping, that is how messenger RNA will get the code from DNA and how it will help in the synthesis of proteins. Okay. Now coming to this, DNA is the genetic material. It has the code. DNA is the genetic material. Okay. It will have the code, because for synthesis of proteins DNA will give the code. Okay, this is the code. That means by seeing the code, that is RNA will proteins will be synthesized. RNA will help in the formation of proteins, but that code, what has to be made? Okay, which type of protein has to be made? So that code will be given by DNA. DNA will give the code and ribosomes will synthesize the proteins. You remember two things. DNA will give the code, that is what type of protein should be synthesized. Okay, and where it will be synthesized? In the ribosomes. Two things, that is DNA will be having the code. Code in the sense for proteins, which protein has to be synthesized. DNA has the code for the synthesis of proteins. Okay, and by taking this code, proteins are synthesized in ribosomes. By taking this code, proteins are synthesized in ribosome. Okay, we'll see the recipe and cook. So the recipe is the DNA, that is the code which is given by the DNA, and then we'll cook the food. That food is nothing but the proteins. What we are making here? Proteins. So DNA has the code, and by taking the code, the proteins are synthesized in ribosomes. Where are ribosomes present? They are present inside the cell. They are present inside the cell. We already know that ribosomes helps in the synthesis of proteins. Ribosomes helps in the synthesis of proteins. Now coming to this, Here this Jacob and Monod, two scientists, they said that there is something, that is the DNA is giving the code and proteins are synthesized, and there is something which is taking the code from DNA and helping in the synthesis of proteins. Jacob and Monod observed that the code is there on the DNA and proteins are synthesized, but there is something in between the that is ribosomes and DNA. There is something which is helping in the synthesis of protein. It is taking the code from DNA and giving, that is taking it to RNA, that is ribosome, so that it can synthesize the proteins. So who discovered this mRNA? This mRNA was discovered by Jacob and Monod. So what they said, that there is something which is transferring the code from DNA to ribosomes, and they named it as mRNA. They named it as mRNA. Now coming to this, how RNA will be synthesized. DNA is the genetic material. RNA has to be synthesized in order to make the protein, in order to synthesize the protein. So if this is the strand, one strand of DNA, that is this one, 3' to 5', if this is the strand of DNA, this strand has the message, this strand has the code. Okay, the single strand has the code, which is, that is code for synthesis of proteins. So what happens is, on this strand of DNA, RNA will be synthesized on this one strand, because DNA is having two strands, two will be separated here, this one strand of DNA which has the sequence, this one, because I said you the strand of DNA is made up of nucleotides. Nucleotides, that is here sugar phosphate will be there, and the bridge is of base pairs. So on the strand of DNA, okay, another, that is a RNA strand will be formed. RNA strand will be formed. So this is the already existing DNA strand which has the code, On the strand, a new RNA will be formed. On the strand, a new strand of RNA will be formed. So it will be, when the new strand is formed, it will be complementary. If it is A always, I said you, A always pairs with T. A always pairs with how will the that is complementary strand? A always pairs with T. A always pairs with T. Here in RNA, the pairing will be different. If this was DNA, it will always pair with T. But in RNA, adenine always pairs with uracil. I already said you, adenine always pairs with uracil. So if this is adenine, it will pair with uracil. Okay. Now if it is thymine, it will pair with adenine. If thymine is there, it will always pair with adenine. But if adenine is there for RNA, if this new has to be joined, so it will always pair with uracil. So DNA always, it has the thymine, so thymine always pairs with adenine. Then again C will always pair with G. A, instead of thymine, here it will be uracil. It will be uracil. Thymine is already there, it will pair with adenine. G is always pairing with C. T is always pairs with A. If A is the adenine, means here in this U, in RNA, it will be paired with uracil. Then C means it will pair with G. G means it will pair with C. So a new strand has been formed. This is the new RNA strand. This is the new RNA strand which is complementary to this DNA strand. So from this single DNA strand which has the code, a new RNA has been formed. A new strand of RNA has been formed, and this strand is complementary to this strand. Complementary means if this here it has G, means it will have, if this is having C, means it will have G. If this is A, means it will always have U. It will complement so that they can pair with each other. So these two strands will be complementary. So this is the actual previous DNA strand. On this DNA strand, a new RNA strand has been formed. A new RNA strand has been synthesized on this DNA. Okay. So whatever it is there, whatever code is there has been translated. So now this new RNA strand, the new RNA strand will be having two parts. That is on this DNA, that is on this, in this DNA now will have two parts. The DNA strand, this is the DNA strand, okay. On this DNA strand, two things will be there. One will be the coding part, okay. On this DNA strand, one will be the coding part and one will be the non-coding part. One will be the coding part and one will be the non-coding part. So this coding part is called as exon and the non-coding part is, so on this DNA we have two parts, that is coding part and non-coding part. This coding part is called as euchromatin and the non-coding part is called as heterochromatin. This on this DNA we have coding part as well as non-coding part. The coding part on this DNA is called as euchromatin and the non-coding part is called as heterochromatin. So like this, one more strand is there, that is RNA's form. So RNA strand will also have some coding parts and some non-coding parts. RNA strand, this is the RNA strand now. So this RNA strand will also have some coding part and non-coding part. So the coding part on this RNA is called as exons. The coding part on RNA is called as exon. The coding part on DNA is euchromatin. The non-coding part on DNA is heterochromatin. Okay, but here on the same one more RNA, new RNA strand has been made. So this strand will also have the coding part and the non-coding part. Okay, so this is the coding part and this black is the non-coding part. Coding part, non-coding part. So like this, on the RNA also coding part will be present as well as non-coding part will also be present. The coding part is called as exon. I have written E1, E2, E3, E4, E5. That is, this is the coding part which is exon. This is first exon, this is the second exon, this is third, another, this is another, this is another. So that is to memorize easily, that is I have given the numbers. This is the coding part, exon part, intron part, non-coding part. Then coding part, exon, then non-coding part, intron. So on this new RNA, we have the coding part as well as the non-coding part. Now what happens is, with the help of endonuclease is a enzyme which helps in cutting. Endonuclease is the enzyme which help in cutting. Now what happens is, cuts will be seen. This will cut. Cut will be made on this. So by the cutting, the introns will be separated and exons will be separated. So with the help of this endonuclease, this enzyme, with the help of this endonuclease, cut will be made. Slicing is done. With the help of this endonuclease, slicing is done. Slicing will be done. Cuts will be made, that is slicing will be done. Okay. So now we have got this coding part. This is the E1. This is the E2. We have cut this intron. We have cut this intron with the help of the endonuclease. We have sliced this one. So now we have this E1, this E2, this E3, this E4, this E5. We have removed that is, they have removed the non-coding parts. Only we have now coding parts. Now these coding parts which were separated, they were at the distance now, they will all 1, 2, 3, 4, 5. So all 5 of them will join together with the help of another enzyme. This enzyme is ligase. Endonuclease is the enzyme which helps in slicing, cutting. whereas ligase is helping in joining the cut pieces. It is helping in joining the cut pieces. Now this is the actual mRNA. So this mRNA has been synthesized from this DNA, on this DNA. mRNA has been synthesized from this, that is mRNA has been synthesized from the DNA. Now this mRNA has the message. mRNA has the message. The message or the code which was on DNA has been translated to this mRNA. So the message which was on this DNA, with the help of this translation, that is this formation of this new one, so has been translated to mRNA. This is the new mRNA. From where it has been formed? It is from this DNA strand only. So now this is the mRNA with all the coding parts. So code is there, so that we can, that is the RNA can synthesize the, that is ribosome can synthesize the protein. Now mRNA has taken the message from DNA. It has taken the message from DNA in the form of code. So now this is mRNA. Okay, and now this RNA with this both introns and exons, that is coding and non-coding part, this RNA is called as hnRNA. It is called as hnRNA, that is heteronuclear RNA. So it has both coding part and non-coding part. So after slicing, non-coding part is removed and we get coding parts, and all the coding parts will be joined together with the help of the ligase. So now this is mRNA. mRNA is very unstable. It is very unstable. To give stability to this mRNA, to give stability to this mRNA, here what happens at this fifth, that is we have this 3' and take it as, so if this 3' here, it will be 5', it will be 3'. So at this 5' end, so this is the 5' end and this is the 3' end. This is the mRNA. This is the newly synthesized mRNA. This is 5' because this is 3' means this will be 5'. So 5' and 3', and it is very unstable. To give stability to this mRNA, two things has to be done. The newly formed mRNA, which is nothing but joined pieces of coded pieces. Okay, so this is very unstable. To give stability to this, we have to do two things. That is, at fifth end, we have to add one cap. Okay, and at the third end, we have to add a tail. So here see this one. This is the mRNA, okay, with the code. This is the mRNA with the code. So to make it stabilize, at this fifth end, we have to add one cap, and to this third end, we have to add one tail. This coding part, okay, is very unstable. This mRNA is very unstable. To give stability to that mRNA, two things has to be done. That is, one, at fifth carbon, that is fifth 5' end, we have to attach one cap, and at 3' end, we have to attach one tail. Okay, that tail is nothing but polyadenine tail. That is, it is poly means many, more than two. Adenine is the nucleotide. So this tail is made up of many adenine molecules. That is, many adenine will combine and form the tail. So it is polyadenine tail, and the cap is methylated guanine. The guanine with a methyl group. So the guanine with methyl group will be attached here, which will be acting as the cap, and this polyadenine tail will be there. So by adding this cap and the tail, this mRNA will be stable. So on this now start, that is coding is there. So the start will be there, start codon will be there, and end codon will be there where the message is ending. So this is the start codon and the end codon. At the start codon, end codon. Okay, the start of the code, the end of the code, that is stop codon will be there. So this is the structure of, stable structure of mRNA. This is the stable structure of mRNA, that is cap, then the coded part, then the tail part. Tail is polyadenine, this is methylated guanine. So this is the coding part. This is the start of the code, start codon. This is the end where code is ended. So that is stop. Stop and start. So this is the actual part which consists of information, that is the structure, that is called as structural gene. So this is how the structure of mRNA is there. So the formation of mRNA from DNA to this mRNA with the message, this whole process, that is DNA will be there, on this DNA a new RNA strand will be made, and this RNA strand will be having all the information, that is coding part as well as non-coding part from the DNA. Then slicing will be done, non-coding parts will be removed, coding parts will be joined together. So that is the mRNA. mRNA is highly unstable. To make it stable, we'll add the cap and the tail. Now this is the actual stable mRNA. So this whole process, so from DNA strand to making of new RNA to this formation of mRNA, this whole process is called as transcription. The whole process is called as transcription. Okay. Now in the next class we will see the structure of tRNA. We have seen how mRNA functions. Now in the next class we will see the structure and how tRNA will transfer the proteins, amino acids.